Hi, everyone. And while some people are still joining, I think we will start. Welcome to our third monthly seminar of the Fish AI Consortium seminar series. You might have heard about the Fish AI Consortium if you are here. So it's an international organization, network of individuals who work together, aim to collaborate to improve and advance artificial intelligence applications in visual analysis for fish species, size, and individual identification. And size is very important, as we will find out today from today's seminar, size identification. So if you are not yet a member of the Fish AI Consortium, you are very welcome to join. It's free, and currently it's all run on a voluntary basis, so we just do it out of enthusiasm. If you are a member, please, it would be great, or even if you are not a member, it would be great if you send me or Catarina Silva, if you send us information about any resources that you would like to share. We have a webpage with resources, and where we link to new papers or whatever models that are available or public data sets and so on. So if you could share more information, so that page will become more and more useful. Maybe you want to promote a new paper that you publish that relates to Fish AI Consortium goals. Also, if you are a member and you would like to see our website to look even cooler and nicer, please contribute some photos, some nice looking photos that we could upload on the website and make it look more sort of slick and nice. And finally, if anybody would like to help with the, organizing the website or our monthly newsletters, which we would like to start sending out, all the help is very welcome. So that's a bit of an introduction to the Fish AI Consortium. I ask you to please, if you are a participant, please just listening here, not presenting, please turn off your cameras, because it sort of reduces the bandwidth and reduces distraction for all the other participants. Just make sure you mute yourself and turn off your camera. Thank you. And now I will introduce our speakers for today. Today it's Tim and Brooke and Jack from the Global Archive. And maybe it's best if you tell more about what is Global Archive.
Awesome. Thanks, Asta. So if my audio breaks up, Jack and Brooke are here to tell me to. But hang on. Can anybody see my slides? Oh, there they go. Whoa.
You might
Okay,
need
yes.
to go.
Sorry.
Sorry.
So
Yep.
we're going to be talking through.
It's coming.
Is it there already?
All good.
Give me a thumbs up if the first slide. Okay, cool. So I'm just going to share a couple of slides about Global Archive and the work we've been doing with that, with fish stereo video annotation. And then Brooke is also then going to show us a video just to talk about some of the needs for AI, and then also share with us one of the quality control tools she's been working on to help work we've been doing with the Australian and other global syntheses of fish annotation data. We're not going to go into too much about what Global Archive is, but essentially it's just a kind of a workflow. Also brings together manuals, annotation guides, and then this quality control work that Brooke's been working on. And then Global Archive is a place for sharing that data, archiving it, but also sharing it and making it available for environmental reporting. But also, of course, a great resource for any machine learning or AI to try and improve the efficiency of fish image annotation. And so we've got mostly data from stereo brubs in it, but also some stereo diver video, and increasingly other kinds of platforms like remotely operated vehicles and benthic observation systems, BOS cameras developed in California. And we've worked a lot with the event measure software, which is sort of the basis of getting that stereo calibrated imagery for getting the length data that Asta was talking about before. And I'll come back onto that a little bit. But, essentially the promise of this fish annotation work has been not only to look at patterns in biogeography, like where fish are, and then maybe give useful indicators for the effects of climate change based on that, but also about the size distribution, as I was saying. So a lot of work being done has demonstrated, the beta video, for example, can be very comparable, the size distribution, the body size distribution, to fishing methods or trapping methods. So the paper's looking at that. And also, various study with an honors project around Rhode Island here, you see that showed that abundance can tell you so much, but it's the length, the body size information that really gives you an idea about the condition of a population, especially under different types of pressures like fishing or habitat change or environmental change. Size distribution is such a key indicator. So a little bit of a story of work we've done in 2018. We brought together about 22,000, it says 20,000, but it's 22,000 RUB deployments around Australia with a variety of partners around Australia. And that was an amazing experience, but it was also an experience in learning that we really have to get better at the quality of the fish annotations and how people do these annotations to make it easier to bring it together, but then also make it better for things like machine learning or AI kind of things. And that study produced some really useful papers that have taught us a lot about conservation planning and different metrics. But really, again, the same message came through from these papers that really it's all about the size information. It's all about getting that good body size distribution data. So for people that aren't from Australia, I was just going to show, I think everybody's from Australia. Anyway, someone from Australia might not watch this or might watch this. So that's a map of the Australian marine parks in the Commonwealth waters outside the coastal waters that you can see. So the green bits are national park zones or no-take zones, IUCN2 areas, but then as you zoom in around Australia, you then sort of see that in the coastal areas, you also typically have some marine parks. This is the Norrie Capes Marine Park in the southwest in the coastal waters. You have the federal waters managed by the federal government, and then coastal waters managed by the state or territory. And within here, you have another set of marine parks. These green bits are sanctuary zones, no-take sanctuary zones. And so one of the things we're really working on is trying to come up with ways to measure the condition of fish outside those sanctuary zones. So Brooke was going to show us a little video from here. But any questions from anybody at the moment, or we'll just keep talking and show videos and have questions later? Is that okay, Asta?
Yeah, maybe let's keep talking. And if people have questions or, you can raise your hand and I
Stick
will
a hand
invite.
up.
Yeah.
Or
You just have to
shove something in
stop
the chat.
sharing, Tim, for me to
Oh
share.
yeah, don't show them the carpet, Stephen said. Okay, sorry. Awesome, lovely. So this is a video that taken in the Geographic Marine Park. We're working with some traditional owners there in collaboration with the National Science Program and Parks Australia to look at the fishes and habitats inside the park. And here you go, zooming in on the Australian marine parks and the Southwest Network and into Geographic Marine Park. And we're in the offshore waters, but in this national park zone that actually links into an adjacent sanctuary zone, which is pretty cool. And so this is the kind of data we're collecting. Here are some fantastic rangers helping us deploy these video cameras off a nice old lobster boat in the marine park. And this is the kind of we're getting. So the promise of machine learning and the promise of AI is to make this more cost efficient in terms of getting the data out of these videos. But what you can see here is you can see, so we're in a protected area here. And so we've got certain big pink snapper rocking up and big jewfish. The jewfish are the ones with the black lines through their eyes. And the interesting thing you see here is quite a range of the size distribution of the species. There's some small jewfish, there's some bigger jewfish. And the most important thing, we reckon, and Asta agrees with me, to get really good condition estimates of these fish populations, we need to capture that size distribution. So any AI or machine learning things we do has to start remembering that we need to understand the size distribution of the fishes, okay? The size distribution of the fishes is going to tell us everything, in terms of of an potentially an absolute condition estimate to be able to assess how well these conservation areas are going. That was it, really. So I was thinking to ask Brooke to do a bit of a demo of, so the lessons we learned from doing the Australian Bruv synthesis taught us about a whole range of, errors that can sometimes happen in people's annotation data. And Brooke, do you want to show us what you got?
Yeah.
And you can, you know the full background, and you can talk to that a bit.
Yeah, it's just loading. Oh, and my video is playing again. Cool. So this is the landing page of Checkem, which is the app that we've made to quality control annotation data. Sorry, what was that, Tim?
No, it's okay. I got the video, but now it's on Checkem. It's all good.
Okay. Cool. So yeah, we learned lots of things from the synthesis data set, and basically we put all of those different ways that people make mistakes into this app and are trying to check so that people don't make them again in the future, or that they can at least flag them and then go back and change them. So first you set the format of the data that you're going to upload. So you can either upload event measure data, so from that program that Tim mentioned, or what we call generic data, which is basically just anything but event measure. And then you choose the type of sampling method that you've used. So we can check single point methods like a BRUV or a BOSS, or transect based methods like a DOVE or a RUV. And then we choose a bunch of other things about how we've annotated it within Event Measure or any other program. And then we choose the directory where we've saved our data and upload. And then while that's uploading, we can choose the information that we're going to check our annotations against. So Checkem has a whole bunch of life history information, like how big a fish gets to or where each fish occurs. So the bioregion that each fish occurs. So because we're in Australia, we have an Australian-based list, but we also have a global-based list. So Australia comes from CSIRO's CAB codes, the codes for Australian aquatic biota, and the Australian marine bioregions, but we've also included a global list which uses fish base and worms. And then now, so our data has loaded in. So this is a campaign or a field trip that we did last year, where this data, we haven't actually cleaned it yet. So now we can go through and check our metadata. And for us, the metadata is the information about where each sample was collected. So where, when, the date, the time, the latitude, longitude, and depth. So a lot of people either forgot to collect that information while they were in the field for the synthesis, or lost it, or it was incorrectly formatted. So Checkem goes through and does a whole bunch of checks. So each of these boxes is a check, and the color of the box indicates to the user if they need to do anything about it. So blue is information, green is it's good to go, yellow or orange is we think it might be an error, or so you should go back and check it for yourself, and red is we think that there's definitely a problem and you need to investigate it. So we also calculate some scores, which I'll come back to in a minute, but each of these boxes you can click and look at what is actually causing the error. Then we have a map which shows all the samples that you've included, and you can see the marine parks as well on there, and each of the deployments you can click on and have a look at the metadata. And so you can look at if there's any samples in your metadata that are missing count, so counts of fish, or if there's any that are missing lengths. So in this one, all of our samples have count data, but there's some that are missing lengths. And then the more interesting stuff is checking the count and the length. So we can look at the total number of fish that we observed in that field trip, if any of the species names have been updated, because taxonomists love to change species names all the time. So we can have a look, and these ones have changed family. We can also look at any species that haven't been observed in that marine region before. So this data is in the northwest of Australia, and according to the life history list that we have, these species don't exist there. And we can also look at any species that just don't match the life history list. So the vocabulary that we're using from CSIRO's CAB codes. Then we can make some plots of the most abundant species, or look at particular species, their distribution across space, or how their abundance changes with different zones, so the habitat protection zones and so on. And then Tim's favorite part is the length data, so we can have a look and see if there's any lengths that are bigger than the listed maximum size on FishBase. So in here we have this species, for example, the max size is 450 mil, but we've recorded one that is 460 mil. So it could be that FishBase just has listed the a max size that's just a little bit too small, or it could be that we've measured a fish that's actually an incorrect measurement. We can also look at the histograms of length. So this is for a particular species, and the red line is the FishBase maximum, and then this is 85% of the FishBase maximum, because sometimes the FishBase maximum is quite extreme. So for tiger sharks, for instance, the maximum length is 7 meters, but if you've measured a tiger shark that's bigger than 5 meters, it's probably also pretty unlikely. So we think that the annotators should be going back and really checking anything that's also in between this 85 to 100% max. In this example as well, we have the FishBase max is just under 300 mil, and there's one all the way up here at almost 1 meter long. So that's most likely wrong. And then we can check that the number that we've counted in each video matches the number that we've measured. So these checks are just looking to see if that maybe you've measured too many fish that were in each sample, or if you have not measured enough fish. And then you can look at each individual sample. So these are the samples across the bottom, or the BRUV deployments. And you can see that the green bits are where the max N has been measured. The blue is where they've been 3D pointed. So you can tell that they're still within the range of the sample. And the pink ones are the ones where they are not measured yet.
So Brooke, can I say some extra stuff here too?
Yeah.
So for me, this bit is a bit of a game changer thingy, because like one thing we did notice when we did the Australian BRUV synthesis is that actually so we have a pretty tight set of guides in our laboratory with the people doing the annotations. But one thing we noticed around Australia is that often people aren't always that great at turning their fish counting into fish, that often the percent measured isn't as many as the counted. And basically, if you're not getting that length data, so if you don't know that length data is accurate to start with, but if you're not getting that length data, you're not even going to be able to calculate any of the kind of condition metrics that we hope are going to come, that are going to be based on that length data. So it's like someone says, I've gone and done a BRUV campaign or a stereo video campaign, but they've only turned 30% of their length of their fish counted into length data. It's actually not, you need to know that. You need to know that efficiency. So anyway, I just wanted to say that it's really important. But hey, Brooke, you can't go back on the previous tab and show us another couple of species with the length distribution stuff? Because that was an interesting one, but you can just look at any species, can't you, from the drop and see, or from the campaign?
Yeah. Yep. So you can choose any species from the campaign, and you can search in this bar as well, and then it will change all the plots below. So it's easy to just scroll through and choose different species that you're interested in. And then there's another one.
Yeah.
There is a question in the chat, and someone is asking whether these annotations are AI generated or human generated, the annotations you're talking about.
Human generated, they are, these ones.
Yeah, so you basically are trying to test the accuracy so that they could be then used for AI training for the proper data set, or whatever measures they could be used for, but also for AI training.
But
Yeah, absolutely.
these outputs could be from an AI tool as well, though. So you could do a laugh test of what the AI is pumping out to make sure there isn't any illogical or unrealistic length measurements that are coming out of the system.
Yeah, we've got a particular campaign at the moment from the Abrolhos Islands where we're running through doing annotations with manually and then run annotations with machine learning with AI, and then we're kind of comparing at the moment how well each one's doing. But the basis of that actually comes down to how well the training data was at the beginning, because the AI is only as good as the training data. So yeah, this is really about sure we have good training data to inform those models.
Can I just interject really quick? I'm just curious, how is the size actually calculated? Like, you have a separate, a human does an annotation, like draws a box around a fish, and then is there an algorithm that calculates how big the fish is? And would the AI kind of do the same thing, or would the AI kind of automatically calculate the size when you're doing the machine learning thing? Thanks.
No worries. Shall I have a go at answering that one? And Brooke might be able to illustrate it. But yeah, so all this data comes out of the event measure software, which is custom made for doing stereo video annotation with calibrated imagery. So it's all from paired video that has been calibrated with a three-dimensional calibration cube. And then in the software, the annotators will typically, for bruvs, they'll typically count the fish, identify the fish, count the fish, see when the frame during the deployment has got the maximum number of individuals in it, measure the fish by clicking on the nose, clicking on the tail across two different windows. And because the imagery is calibrated, that'll then tell us the three-dimensional positions of those noses and tail annotations to then calculate the length. So it's all done from proper stereo calibrated imagery. And what we're working on at the moment with these nice guys from the AFID initiative is to bring machine learning into that, especially to do with the identification of where the nose and the tail are of the fishes, and then let the actual stereo library that's already existing in the event measure software do the length measurement, because the length measurement bit is kind of easy. It's done with the stereo calibrated imagery. The tricky bit is identifying where the noses and the tails are of the fishes. That's what the humans have to do. We've already kind of got the machine doing the measurement bit. It's more about identifying where the nose and tail is, and whether it's the right fish or not. But yeah, no. So it's all done through event measure software. Is that a good enough answer?
That was perfect, Tim.
Yeah, thank you. That was really, really good. Thank you.
Sweet, no worries. Yeah, so because that stereo calibration stuff already exists, the actual generating the length, we don't need to use a machine learning to try and, hammer out our estimate of the length, because it's actually, as long as you collect stereo calibrated imagery, the stereo bit's easy. The hard bit is the nose and the tail. So that's what we're trying to train at the moment. Some models to consistently identify the nose and the tail, and then track those fish through time to get repeated measurements and stuff of the fish. Right, I better hand back to Brooke, eh? I'll shut up now.
Cool. And then we can also turn the length into biomass using the length weight equations that are on FishBase. And so this here is just a plot of the top species by biomass. And then we can look at the biomass across space as well. And then lastly, you can download a list of all the potential errors within your data set that you've uploaded. And you can also download a tidied, clean version where you filter out some of the errors. But we do think that it's best to go back to the annotations and make the changes to the raw annotations instead of just filtering out from the data set, so that you maintain that connection between your annotations and your raw data set. And then this is the quality control score plot or beer coaster that we've made. So each of the pies, the different colored pies, is a different quality control score or metric. So the green one is metadata, and the solid line is the goal that we think that people should be aiming for, and the fill or the number is how well that campaign or data set did for each of those scores. So for metadata, it's how much of your metadata meets the right format. So in this example, 100% of the metadata met the format. Count schema is how many of the species that you've seen match the schema or the vocabulary that you've chosen. So almost 90% were included in the life history list. And then these ones look at different ways of, have you measured all the fish that you've seen? So did you have too many measurements or not enough measurements? And have you seen any fish that are bigger than their maximum size on FishBase? And these ones here with the asterisks are for large-bodied carnivores. So they're ones that are really important for looking at the condition of the marine parks and other areas that we look at. And Tim, did you want to talk any more about the large-bodied carnivores metrics?
How do you know? But yeah, like so this is cool in the sense that now what I'm really enjoying with what Brooke's been doing here is we're getting a nice way of being able to just in a quick in a nutshell see how well people have annotated their stereo video data. And so I'd say this is looking, so why is that bit of pie not good, Brooke? Again, the red one?
Oh yes, sorry. So this one is just under half because we hadn't actually finished measuring this campaign. So we had counted all of the fish, but we hadn't measured all of them. So just under half were measured.
Okay, cool. So when you've got a finished campaign and you put it through something like this is a really simple, quick graphic, or even we output this outputs as well as like a little CSV with all the scores for each individual thing. You want to know this video has been annotated well before you use it for training. That's what I'm going to argue, I think. And like, you want to know it's got a good quality control score before you use it for training, and whether it's then going to help you in the mission you might have, which is what our mission is, to try and increase the amount of length data and the sensitivity and the robustness of condition estimates based off the length data. Yeah, and so the reason we have that asterisk there for those particular fishes is we did also notice that there's a bit of a tendency of people to annotate some fishes better than others. And so, yeah, it's really important to know that you're actually capturing those ones that you're more likely to end up using for certain condition estimates. So like often gets used a lot for like climate change, tropicalization type indices and stuff, but it's often those sort of larger bodied fishes, the larger body carnivorous fishes that often might be more useful for even some of the habitat metrics we've come up for impacts of changing habitat, but then also some of the impacts about different pressures such as, inside, outside, protected areas or not. It's really those fishes that end up being used in those metrics. So you want to know that people have done a good job in annotating and generating the length data for those species, because otherwise because those metrics are all terribly, the ones that we've seen so far are very, very hungry for data, in the sense that the more length data you have, the more powerful the metrics are to discern what's going on. So that's pretty cool, isn't it? And I suppose the other thing to say is that one of the development plans we have for Global Archive is that this QAQC tool will essentially be the gateway to be able to put the data into Global Archive. So before you can share it and before you can QAQC and have your QAQC scores recorded against your data set. But that's kind of that's currently under development at the moment. Is that all right?
Yep. That was all I had for the demo. Did you have any other slides, Tim?
No, that's all my slides, but do you want to open up to questions and things now, Asta?
Yes, sounds good. Thanks a lot for this presentation. I think, Brooke, you can almost leave the screen on because we might have some questions related to this.
Yeah, and we haven't mentioned, hey Brooke, you haven't mentioned as well that this is all available as an R package that Brooke's published at the moment, but Brooke's working on a publication about this as an R package and this kind of thing. So it's all available on the web, free to use.
Open source.
So, yeah,
that was
my first question. So if I wanted to check some data set, I mean, in theory, this can be used for any So what this would be, so for any data set that would have species and sizes in them for a marine species, right? Not freshwater, because you wouldn't have freshwater.
You could put it in, yeah. You could put
And
it through.
basically,
It's designed to work with event measure data table outputs as well, but it also has generic format, so you can put any form of data, you can format any kind of data to go through it.
yeah.
Yeah.
So basically it will help me to say if the data set has some lengths that doesn't seem to be realistic, and if the species seem to occur outside its expected distribution area. Because, for example, global information, bioinformation, or GIF or GBIF dataset, for example, that I think a lot of, iNaturalist and so on contribute the data there. I think doesn't have size data, but even the species occurrence data can be difficult there sometimes, and so presumably you should be able to use that as well, right? Is that
Yeah, absolutely. One issue we've seen is that sometimes people that get trained in fish identification on one side of Australia tend to kind of see those species more often. And so you get like East Coast fiddler rays turning up in the West Coast, that kind of thing.
Yeah, that's right. But also, so we try to, Steve, do you want to ask the question? I don't know if you can turn unmute and ask it. If you want to be able to.
Who, me?
Yep.
Yeah, I was just curious. You mentioned the package will be open source in R, but if an organization puts in data, can any other organization then get that? Say, for example, WA government puts in a bunch of data, all the universities can then access it. Similarly, if Queensland puts in data, we can pull it and do a paper or a collaboration. Like, is that the plan ultimately?
Oh yeah, sorry, I should have given a much better explanation of Global Archive. So Global Archive was built as a repository archive for stereo video data, but it was also built from the beginning to kind of cope with that question that you have, which is that that not all data sets can be shared openly, or in fact should be shared openly, I would argue actually. So what Global Archive has is, as a standard, the metadata is discoverable by everybody. It's publicly available and sort of just out there as a BYCC kind of thing, so that the sample points become discoverable in the platform. But then the actual annotation sets or the count or the generic count length data you upload, you can keep that private, or you can share it with individual users within Global Archive. Within projects, you can share a project with individual users, and then they can then access it and share it amongst people, or you can make it openly accessible. And so the idea for that is that a lot of the work we do for the Australian government, they insist that it's openly discoverable as open data, so we make it open. But then other data sets, like ones we collect for industry, or ones that collected with government departments, or even or actually particularly at the moment, work we're doing with traditional owners, where they might be mapping particular cultural features with BRUVs or some other kind of method. They don't want those data points out there, so we're working out a way of keeping those data points secure for them, but at the same time enabling them to share it with someone like Parks Australia to use it for environmental reporting. So yeah, Global Archive is effectively designed for that. But yeah, sorry, I didn't think to go through that kind of a thing in this, but yeah, good
so
question.
Maybe just to clarify that the package is freely available. This is just an R package to test any data sets, right? Something that you developed as a side project of Global Archive, or which is now an important part of it.
Yeah. Well, and I think part of the workflows we're just trying to put out there, trying to make data sets better and in general. But then, in the future, we're hoping to sort of wrap that in a bit into the Global Archive platform as well, which is very much designed about making data discoverable, but not necessarily open, unless it has to be open or needs to be open.
So a question about open data, isn't there a risk, and I suppose that's what you say, not all data maybe should be open, is that if we put all this fish data open, does that also invite, illegal fishing and other issues to high quality sites?
yes, whenever anybody says that all data, fish data should be open, I say, yes, I believe that Geosciences Australia map of gold should be open as well, but they refuse to make it open to me. I don't know why. But anyway,
Yeah.
so yes, I think I agree. But at the same time, for national environmental reporting, for state environmental reporting, you know, data sets should be able to be contributed. So it's really important to come up with a way where you can have a trusted way to share confidential data sets towards general syntheses projects. And that's kind of the thing that Global Archive's designed to sort of help with. But yeah, no, I wasn't going to talk about that, but maybe I should have done. Sorry.
That's all good. I had a question for Brooke, though. And everyone else is feel free to, raise your hand or ask questions. So what about, because we tried to do something similar as well with different data sets, and it's always a challenge to decide what length data we should use as a benchmark. So if a fish is longer than fish-based lengths, like, how did you decide? Because I find that fish-based lengths can be either too small or too big, and that's kind of a challenge. How do we decide that maximum length?
Yeah, so usually we look at the data set as a whole, and then we'll always go back to the videos and look at the actual measurement that we've taken, and we'll try, if it's been flagged as that it's too big, we'll try and measure it again. But most often you can kind of tell if it's just like ridiculously too big, that you've measured it, or if the fish base maximum is wrong. And so usually you'll get a campaign where you've gone through and you've done all the measurements, and you'll see that there's like 100 that are over the fish base maximum length, but they're all very consistent sizes. And so then we kind of know that it's actually the fish base maximum length that's wrong, well, that we think is wrong. And so what we've done in Checkham is we've made this edit maximum lengths. So we can add a new maximum length for a species and then include a reference information, so like a website or a paper that lists the new maximum size. And then eventually our plan is to then give that list of all the species that we've updated the maximum length for and give it to Fishbase so that they can edit their maximum lengths.
That was my next question. Are you going to share it with Fishbase? Because I know that they really want to improve, their information. Yeah. So yes, yeah.
Yeah.
Okay, another question I had is that, so a lot of people here on this Fish AI Consortium, want to use good quality data sets. So you have a lot of underwater video data sets that could be used for training species recognition models, or maybe size recognition models, or even individual. Like, imagine if you could identify each individual fish, like separate as we would identify humans. So is that accessible, and how would that be available?
Yeah, so we worked with AIMS, the Australian Institute of Marine Science, a few years ago to make this OzFish dataset, which they set up as a challenge for a fish AI training challenge they set up a while ago. So we made a whole load of data from that we collected around the northwest of Australia, the imagery and the annotations available via GitHub repository actually, and yeah, so there's that, but then at the same time there's heaps of other data sets around Australia that would be. The problem with that data set we shared to the OzFish thing is that it was from the northwest of Australia, and a lot of it we were kind of really interested at the time in looking at recruitment dynamics in a couple of the really important commercial species there. And so we ended up with an awful lot of annotations of one particular species. So the people that use that data set came up with some incredible models for one particular Lathrinid species, which is awesome. But I didn't have much great training data for the rest of it. But yeah, like we're using a data set at the moment with the AFID guys that are working with Event Measure from the Abrolhos, and one of the reasons we're using that, actually, it has a really nice selection of the main kind of key target species for the fisheries, but also the visibility is really good, and it's like making it definitely easier, I reckon, for the machine to do a bit of learning on that imagery. But yeah, no, we've got heaps of data, and all the stuff we've been doing recently for Parks Australia through the National Science Program all around the northwest and the southwest is all publicly available imagery and annotations. So it should be a great resource for training stuff.
Yeah, excellent.
I think the important thing just to add there is videos are large. So we do store them. IMAS, for example, has a repository which we contribute all our images into, and increasingly making them more and more open to make it accessible to others. But it is still a bottleneck that you've got the annotation data, that's all there, really accessible. The video that sits with it is still links maintained back to that annotation data, thanks for things like Event Measure. But the video itself is a bit more of a request and have a chat to us sort of thing.
Yeah.
Your interest levels are, just because, yeah.
With the Ozfish one, hey Jack, we did a stills capture, so like most of the machine
I think it's breaking,
AI
Tim.
stuff people are doing points, but
yeah,
it's definitely a. Sorry, I broke.
Yeah, you broke up a little bit.
Tim is
I
joining from a farm in Western Australia, so yes, we are still struggling with his internet.
Okay,
yes.
I broke. I'll stop talking. Bye.
No, don't disappear just yet. But I can see Scott has a question. Hi, Scott. You can turn your camera if you like.
Oh, well, let's see. Oh, yeah, there we go. Hi. My camera's on the wrong side of the screen. Okay, hold on. Thank you guys so much. That was really, really cool. It's really nice to see this platform and just see what it does. Really, really powerful. My question, I guess, is I've been talking to people about, I guess there's some work being done to put cameras on fishing boats and apply some of this machine learning kind of stuff to doing, I guess, similar stuff like species ID and potentially measuring fish. And I'm just curious if that's something, if you'll be integrating data like that as it starts to come out, because I can imagine having both the underwater data, but also the data on the fish that are coming out of the water. Having both would be pretty cool all in one place.
I think my first reply to that one, Scott, is if it's available publicly, then yes. A lot of that stuff has fairly strong sensitivities around it, so it is a challenge to I guess bring that data to the table before you can even navigate how to contribute that into the system. But there's nothing stopping the system from ingesting that kind of data stream.
Yeah, nothing from putting it into Global Archive as a private data set either, because I mean there'd be no difference in the annotation data, it'd just be the same, wouldn't it? But yeah, no, cool. I know there've been people working on crustacean and fish stereo systems on fishing boats for crustaceans and fish recently. That sounds really, really cool. And I think it's about capturing that stereo calibrated imagery, because that's the way to get the robust length data. That's going to be transferable across species and across, well, you can measure anything with it. But yeah, no, cool. Sounds good.
Cool, thanks.
So does Global Archive, do you only want data with stereo calibrated imagery? What about some recreational fishing data where, let's say, anglers are posing with the photos of fish that they caught?
Well, there's no reason why we couldn't do that, Asta, because I was looking at your paper before on that, and it does look very cool. So yeah, totally. Good point. There's no reason why we couldn't do that. And I think I just have a strong bias to the stereo video thing.
Yeah, I think Tim
Sorry.
made Global Archive and Checkham intentionally flexible so that it could theoretically take a lot of different types of data.
Yeah, I think it's always about what is going to become the main platform, and there won't be probably one main platform, and that's all right. I mean, like, why should one platform become the main? But it's great if these, if data sets can be shared. That's the biggest issue, I think, now. What hinders AI development, right, is this availability of the training, high quality training data sets. And a lot of people here in this consortium are frustrated about it and hunting for them and spend a lot of time of cleaning and annotating and sorting data sets and making sure they are correct.
Yeah, and I think that's the important thing you touch on there, Asta. There won't be one. The globe's too big. There's too many institutes developing their own thing, right? But as long as things are interoperable, meaning that they can talk to each other, and built around some of the standards like the OGC layers for the spatial layers and stuff like that, then it makes the job of bringing together data to standardize it even more simple. The other one there is being able to check and standardize vocabularies coming out of these systems so that they are in a standard, that it's not in old man up the road calling duck, boarfish, duckfish, and being able to have to manage, that vocabularies around the place. So
There is a question from Peter Mitchell in
No,
the chat, maybe.
take it out of the chat.
All right
Is
then. Yes, I'm just having a look at this check EM, and there's a sample metadata. Like there's an example spreadsheet, but it's like currently blank. Is there an indication of which fields are mandatory to be able to sort of so that we can so I can force something through and see how we did?
Yeah, so there's on the left, there's a user guide down here, and there's also one up here, which downloads it. And then if you go to that, there is a table column requirements for the metadata, and then I've got which ones are required, and then the format of each of them.
Perfect. All right. Fingers crossed. I'll see how complete our one was. I'm guessing
It's been tested on Tim and I, Pete, so it's a pretty low bar. We've managed to get stuff in, so surely you can manage it.
No, I was saying how complete the metadata is.
Yeah, and your data, Peter, will look 100% on everything.
Wow. So that actually, so on the length measurements, like on the recent project we've finished, we just focused on like key fishery species. The time taken to measure everything would have just been sort of exhaustive. So now Tim's saying
You're
like
talking to people that have done it their whole lifetimes, Pete. What are you saying?
You look exhausted?
Yeah, I'm exhausted, totally. Yeah, knackered.
Yeah. So when you were saying like if you're not sort of completing that, then you're really sort of you're not capturing all that important information. Do you mean it's really important to capture length measurements of everything at all sites, or like if something has to give, what would you recommend?
No, I think that's where that's one with the star on kind of comes from, Pete, because like over in New South Wales, for example, the New South Wales DPI guys, they only really measure their key target species, and the most of their stuff is for inside outside sanctuary zone stuff, but they're mostly measuring the species they think are likely to respond inside outside sanctuary zone. So they would do pretty well probably on that asterisk segment of the pie, whereas they would do very poorly on the ones for the rest of the assemblage. So I think I think probably what you're saying is pretty logical and rational, to be perfectly honest. And yeah, I haven't it's a bit of a bigger chat really, and I think we need to actually do some papers and studies to demonstrate where the really important what those important species are to get the length measures of. But we've kind of come up with a definition of large body generalist carnivores, which pretty much covers the species you're probably talking about that you measured, I'm guessing. Are you talking about pink snapper and Coburn Sound and stuff?
Yeah, trevally, some of the whitings, just whatever. And the elasma breaks, whatever we considered of some fisheries importance. It doesn't have to be like massive.
Can I ask a question here though? I'm always confused. So is that measurement? So are you talking about Brock data, Peter?
Yeah.
So is that measurement done only on max n, which is like a screen, which is the capture, or like a second or whatever, however long, where you have a largest number of fish? Not everybody might know what that max n, a largest number of fish in the screen, right?
Of that species.
Of that species, right? And if that is true, is it so that we would be more likely to underestimate big fish, because when the big fish comes into the screen, all the small ones disappear, and that doesn't become a max n? So you will have max n only when there are more smaller fish.
Yes, there are problems with it, Asta. That's correct.
So how do we get the full size distribution? Do you need to measure at different times? And I guess this is what we are working on.
So this is a project we're working on at the moment with the machine learning AI guys, because I had a really nice honors student who worked on this a while ago, but it almost sent her crazy because I tried to get her to measure the fish all the way through the deployment. You know Amy Coughlin?
Yeah, I know. Yes.
Okay.
She finished PhD with me, yes.
Yeah, okay. So she didn't go crazy, but it was a really big thing to ask her to do in her honors, was to measure all these fish all throughout the deployment. And we saw some really interesting patterns, some really interesting evidence of biases you get, especially for assessments inside outside protected areas, especially for things like pink snapper, which are very mobile. Really interesting biases in the data. And so, yes, there are definitely some issues that need to be resolved. But what we're trying to do now is use some machine learning AI things to do it, because Amy had a huge job to try and do it for just a couple of species throughout these hour-long deployments that are standard for bruvs, stereo bruvs. And so, yeah, no, there's definitely, this method definitely does need some work to refine that issue you talked about, because there are some biases, I think, in that use of a max n, which is the maximum number of that species at one time on the video. And but it's actually, I don't know if you're consistently always getting the big ones and always missing the small ones. Sometimes it seems the other way as well.
No, I
So
meant the
don't
other
know.
way. So you are more
Oh yeah,
likely
no.
to
So
get
well,
small
yeah.
ones
So
because,
what?
yeah.
Sometimes it seems one way, sometimes it seems the other way. It seems that from what I've actually seen, more likely we seem to be missing more of the smaller ones than the large ones. We seem to get the big ones, but often miss the small ones. Sometimes a bit in some of the stuff. We've done some comparisons between baited and unbaited, which have revealed some interesting patterns and stuff. And yeah, but it is a standard. It has been used pretty consistently around the country and internationally. So it's at least a start. But I kind of think we need, that's actually where I think we need machine learning and AI. We need it, especially in those places where we can't send our research assistant and honor students mad every year, asking them to do really horrible, where the machine can happily do those annotations that are repeated throughout the deployment. You can't ask a research assistant to do it because they just go crazy.
Yeah.
But yeah, to get those metrics out of that, we need some AI, I think.
And we need collaborations and more research to test the comparability of them. So we are approaching 1 hour mark, and I know some people might want to leave, but there is still one question from Scott in maybe the last question, and from Scott in the chat, if you want to say it, Scott.
Yeah, just curious about other organisms like seaweeds and stuff like that. Are those at species level in the annotations? Just non-fish?
So we've done a lot of work with Jack, particularly with annotating the field of view from the bruvs of the benthic epibiota. And I suppose what we've generally ended up using the Katami schema, which is more for species kind of a thing and doesn't really go to species except for a couple of things like we go to species like for Aclonia radiata, which is the most common macroalgal, subtidal macroalgal species in southern Australia, and go to species for Aclonia radiata and another couple of algae like Scardathalia and things like that. But all the sessile inverts and everything else is just morphological types.
But it also starts extending across into something like the Squidle Plus and understanding marine imagery, IMOS investments, where we are looking at more of the habitat end of the spectrum rather than a covariate to the fish side of things, which is where we sort of focused our efforts.
Yeah, but you still don't get much far. You still can't get far into the species unless you got a divining rod.
No,
right.
genus and stuff like that. For corals, you can get down to that. We're starting to get there.
And I guess if somebody was interested in like a specific thing, they could always go back to the data and add additional annotate. Like if somebody's really interested in a specific kind of algae or something, they can go back and try to identify that and add annotations to things.
Yeah. I mean, Hamish Malcolm did that with Adriano Verges on the north in New South Wales. They came up with a study looking at change in kelp forests after a heat wave, and they went back and kind of re-annotated their kelp in the video, in their stereo video stuff. And we've done a bit of stuff on the Northwest Shelf recently where we went and re-annotated. We went back to the videos from a big trawl closure area and went and measured all the sponges and stuff and found out the sponges were taller in the trawl closure than they were in the trawled area, which is a massive surprise. But you can definitely, that's the wonderful thing about the video, I suppose, isn't it? You can go and reuse it.
Well, and that goes back to what Asta was just saying before, where you can unpack some of these biases. You can actually unpack them because you've got the permanent record of video there. You can go, oh, Maxine is biased towards for this species or in this area to the bigger individuals. Reef Life Survey or the diver observed stuff. The only point of truth is the data sheet that was captured in the field, and you can't really start unpacking some of those inherent biases that the bigger fish hang just outside the field of view of the diver, and you never see them. They go unrecorded, and you can't challenge that kind of data set. So it really does smash home the value of curated and persistent video archives or imagery archives.
I have to put in some defense for Reef Life Survey. I think there are still ways to assess biases, because also with the video we don't know whether it was just not recorded, if it's not in the video, if it's not coming. But I agree, like different data sets have their strengths and weaknesses. It would be great to actually do some comparisons and just really start looking at their selectivities and biases, and that's where we try to move into that direction.
Yeah, it'd be nice if your biases were hidden, though, more hidden, though, wouldn't they be? No, sorry, I shouldn't say that. Sorry.
All right, okay.
Someone's going to come back and
thanks.
reanalyze some of our videos, Jack, later on. They're going to go, oh, they had it all wrong. And it'll be like, oh, Ruth. Oh, at least we got a paper out of it. No.
Well,
sorry,
that's how science
say that.
develops anyway. Like,
Yes.
we say someone
Then they
was
can- then-
wrong, and yes, we can
And then they'll
improve it.
cite us. Then they'll cite us at least. There'll be one person. That'd be good.
All right, thanks a lot. And Tim and Brooke and Jack, this was a very great presentation.